So Senya is a new company. We are focused on this very exciting target called LARGE1. LARGE1 is a target that John has been working on for the last 2 decades. It's an important target because it is implicated in a number of chronic diseases. And we're very excited to be the leading company in this space. Okay. Just by introduction, so I've been in the industry for about 30 years. I was working for McKinsey for 20 years, where I was a partner serving pharmaceutical companies. Then I joined Shionogi and then Arena Pharmaceuticals, where I was head of Europe and head of international. Since 6 years ago, I've been working with Oxford University and UCL in London to take breakthrough technologies into the clinic. I met John 3 years ago, and I joined the company because I was very much excited about this target. Okay. Together with me is Chris Martin. He was the founder CEO of Spirogen, which he exited to AstraZeneca. Since then, he's been CEO and chairman of a number of biotechs. And we're very happy to have him in the executive, non-exec board. Okay. But to you.
Yeah. So just by way of introduction, I'm John Green, one of the scientific founders of the company, alongside Steve Moss. Steve's now retired from university, but very much engaged in Senya Therapeutics. I'm still just about a professor at University College London, where we discovered the target, but now spend most of my time with Senya Therapeutics. We've built a remarkably strong group of advisors, key opinion leaders, for example, in nephrology with Professor Dwan at Edinburgh, Dr. Steve McAdoo, Imperial, Professor David Wheeler at UCL. Also in cancer, for example, we've tied up with Christian Blank at the Netherlands Cancer Institute and mentioned that a little bit. And we also collaborate with numerous very top university laboratories around the world, including in New York, Heidelberg, Tokyo, and several others in Italy, Amsterdam, and in the UK. So we're in a position now where we really are sort of very much ready to move this into the clinic.
Now we'd like to talk about LARGE1. It's a very interesting target, just coming out of an age. Okay.
So what is the target? It's leucine-rich alpha-2 glycoprotein 1, or LARGE1. Many of you might not have heard of it, but I can guarantee you will hear about it a lot more. As you can see from the graph on the right, the interest is growing exponentially. It seems to be pan-disease insofar as that it's involved in many diseases. It's induced and expressed, and it destabilizes tissue. It causes dedifferentiation of cells, and as a consequence, this promotes the disease progression, whether that be in lung disease, kidney disease, eye disease, or cancer. One of its major mechanisms is through modifying TGF-beta signaling, causing a TGF-beta signaling switch. And it's important to add that this is upstream, very much upstream. So it's induced, and when it's induced, it causes disruption and disturbance of TGF-beta signaling. And as a consequence, you can imagine it's involved in many, many diseases because of this.
So this purple bit is LARGE1. As you can see, it actually latches onto this TGF-beta receptor and locks that in. It's the problem.
Alters the configuration. So we've been involved in publishing numerous papers. It started off with a paper in Nature back in 2013, and subsequently in papers in Med, Science Translational Medicine. In fact, we've got another paper in Science Translational Medicine coming out in about 2 weeks' time. We're entering the IND enabling stage. We have our lead antibody with very high affinity for target. We've done a little bit of NHP work showing that there's no adverse response in NHPs with systemic dose. In fact, the knockout mouse is perfectly normal. So we have very good safety prospects. And we are just finalizing some further optimization of our lead antibody, and this will be completed by the end of the year.
And much of this work has been funded by the UK government itself, so we have been quite lucky with non-dilutive funding. So in terms of programs, because of this target being pleiotropic and can be addressing quite a number of diseases, we have choices, but we have decided to focus on kidney because this is where we can see that the large one is a major driver of the pathological process, but also here is a quite high level of unmet need. So this is our lead program. We also have an early stage preclinical program on lung cancer and retinal disease. The plan is we'll push the CKD all the way to end of phase 2 in the next phase. For the other programs, we're looking for partners to develop. And as resources become available, we like to expand the reach of our program. Okay. Now we talk about kidney.
Yeah, so very quickly, because I can see we've only got 3.5 minutes left.
Yeah.
As I mentioned, it's involved in disturbing many sort of cell types because most cell types express TGF-beta receptors. And in the kidney, it's a very complex organ, for example, 40-odd cell types. But key players such as the tubular epithelial cells are dedifferentiated with large 1, apoptosis occurs, EMT. It disturbs the vasculature and then promotes a later stage neovascularization. And critically for chronic kidney disease, it drives myofibroblast formation and also alters the infiltrates of the immune system. So as a consequence of this, it's not surprising it affects kidney function. Interestingly, there is a SNP in the promoter region of large 1 that's been identified in diabetic patients that leads to enhanced circulating levels, enhanced induction of large 1 expression. And this correlates with those diabetic kidney patients with the worst prognosis. And we've commissioned some work with colleagues at Imperial in the UK and Edinburgh in Scotland. And they've shown that in archival material, both in plasma or indeed in urine, there is a really strong correlation between circulating large 1 and loss of kidney function. And this indeed correlates with the stage of disease. And there was great concordance between the two sites, showing almost identical data. And we have further data to show. Skip very quickly. This really just confirms the animal data, which shows that if you knock large 1 out, you actually improve kidney function in models of the disease.
And we're just completing a preclinical studies, and then we'll start the clinical studies focusing on patients with stage 3B or plus kidney disease. The goal is to prove that our drug can actually prolong the time to dialysis by more than 6 months, and we're pretty confident that will be the case.
Very quickly, it's involved in cancer as one of the other indications we're interested in. It affects many of the aspects of cancer, importantly, not only the primary, but also it affects the metastatic niche and promotes metastases. Our colleague, we were contacted by Christian Blank from the Netherlands Cancer Institute because he found in an unbiased analysis of serum from patients that were non-responsive to checkpoint inhibitors in melanoma. He was trying to find whether there was anything that correlated with that, and he found, lo and behold, large 1 was the marker that actually correlated with poor response, 25% of patients who responded poorly. In fact, he's now added low PD ligand 1 as a biomarker, and he said it's the most highly predictive markers for poor response. So we actually have a great biomarker for going forward into patients and stratifying them.
So this is an opportunity for precision medicine for treatment-resistant patients.
And it's important to add, actually, the Netherlands Cancer Institute are so interested in this that they've invested in the company, which is quite unusual.
So we have IP protection up until 2050 for both programs. And as far as large 1 space is concerned, we can see that we have no competitor at this moment. Okay. So what we're doing now is we're raising the next year Series A to cover the kidney program. We're also looking for R&D collaborators on other programs. So, please look up large 1, and if you're interested, please let's talk. Thank you.
Thank you.